biopesticide or whatever. I don't know whether you want to comment on that, or maybe Antje could discuss a comment on that because of the OECD meeting. So Vera first, and then maybe Antje could say what the feeling is.
I am not an expert of regulatory aspect, so I leave the word to Antje, which is more knowledgeable about these issues. It will depend on the regulatory bag in which they will fall, as I was saying also Hilde before. So Antje, if you want to add something.
Yeah, I can also add, it depends, of course, on the regulatory regimes. And I think there is a clear regulation, as I already outlined. And I think what Michel maybe refers to is the special case of virus or RNAi for viral defense. And he says there are no pesticides against viruses. So maybe we can see it as a kind of vaccination. But on the other hand, if you apply dsRNA for as a sort of plant protection product, then it's considered to be a plant protection product. And as I said, as it is currently, there is no specific category where a dsRNA may belong to. It's just considered in the moment as a conventional pesticide, but this may change in the future, I don't know. And and on the other hand, I think if the dsRNA is expressed in the plant, of course, it's a GMO. So I think there are clear regulatory requirements for both cases.
Yes, I think in the USA, Canada, it's classed as a pesticide or a biopesticide. And I think that's what Nina was saying, Nina Mitter was saying about Australia, that it would be classed as an RNAi, as a biopesticide. Okay, I see that Bruno's now activated the recording.
I'm sorry.
So unfortunately,
Mr. Hilde, you need to repeat your presentation. But sorry.
Well, you didn't have your espresso at lunchtime. That's
yes.
Okay,
so Marco said, remember me. But now it's now recording. So please go ahead.
Okay, now we move on with the program. And there was a special webinar organized by the Honey Sweet Consortium to talk about the Honey Sweet developments and application. And Ralph Scorza from USA is going to talk about that for us. So are you ready, Ralph? Over
yes, I'm ready. I'll try this screen share. Let's see if I can find my video here or my PowerPoint. Let's see.
I hope it wasn't too much of a rude awakening for you today.
No, it was okay. It was great. I'm usually up early. Okay, so we'll get rid of this. Okay, so I was everyone can hear me, I assume?
We don't see your screen, Ralph.
Oh, you don't see my screen? Oh, let's see.
Screen share again.
Share screen. Oh my goodness. How do I? Okay. Oh.
You do the share screen at the bottom and then
share screen, right? You see anything now?
No. Then you need to open and go on your PowerPoint presentation.
Okay, I'm on PowerPoint now.
Yeah, when you do the share screen, you need to click on your PowerPoint.
Okay, so
share screen and then click on the PowerPoint, which should show.
Okay, so share screen and click on the PowerPoint. Well, let's see. Is that, you see it now?
No, we don't see it now.
Oh my goodness. Okay, so let me try again. Share screen.
Share screen, and then it should show what's on your screen, including the PowerPoint, and you click on the PowerPoint.
When you click on share screen, there should be a pop-up window with different windows you have open, and then you have to select
share.
Yeah.
Open system. It says open system preferences. I don't know what's happening here.
You have your PowerPoint ready? It should just.
Oh yeah, it's ready. It's on, and I can see it. It comes up. It comes up. Well,
What
maybe.
What I have experienced on my computer is that I have to search my computer and then I find the PowerPoint presentation and I click on that one.
Okay, so I have.
If
All right, let me go here. Let's try this. Okay, try it again. I'll hit the share screen, and now I'm going to go to the PowerPoint. That work?
No.
Why
Okay.
don't we go with the next speaker, and maybe Ralph, you send me by email your presentation and I will share for you.
Okay, let's try that.
Yeah.
Send it to you in an email?
Yes.
Okay, I'll give it a try. Sorry about that.
I don't know how long it takes also by email. I don't know if we want to go on with the next speakers.
Well, the next one is Hughes.
No, okay. No, let's try to finish it.
Okay, well, oh gee, I don't know what, I have my PowerPoint there. Here it is. Try one more time. Here it is. Open, it's open now. And now let me try share screen with it already open. No, I'm sorry. It's just, well, what I think maybe what I'll do is just do it without without the PowerPoint, okay? And I'll be able to look at the PowerPoint, but you all won't be able to see it, unfortunately. So here we go. Okay, so I was really, really impressed with the communication talks that were given today. And this is where we are with the Honey Sweet situation. There's a large group of collaborators that have been working on Honey Sweet, the development and the evaluation and the testing for 25 years now. And this project started in the early 1990s. In the mid 1990s, the project went into field testing in Europe. So we have a number of people, and again, I can't show you everyone, but the first speaker that we had was Dr. Ravelli-Nandro. He talked about the need for plumpox virus resistance, the symptoms of the disease, fruit deformation, premature fruit drop, poor fruit quality, tree decline in severe cases of infection or susceptibility. He went on to discuss the problem with the virus in countries and the great losses of production in plums. Now, the virus affects all stone fruits, plums probably the worst. And he went through the aspect of resistance, that there are no high levels of resistance, certainly in plum germplasm. And the real way that we're working on, or the EU is working on containing this disease, is through quarantine. And I think we all know that the quarantine is not entirely effective in many cases. So the virus continues to spread. Why is this a problem? Well, of course, we have a reduced fruit quality and reduced availability of a very nutritious fruit for consumers. And I've been to some places in Europe and seen some of the infected fruit in the markets, and they really are neither very appealing many times or or the taste is not the best because of the virus. So how this project started was we were afraid that the virus might come into the United States, and we wanted to see if we could find a source of resistance. And we used the plumpox virus coprotein gene in the same vector that Dennis Gonzalez used in the development of the papaya ring spot virus resistant papaya, which has been approved in several different countries at least. So we use that, and that was basically the insert of the coprotein gene. But what happened was unintentionally that on the insertion in one of the plum plants, there was a formation of a hairpin. Now this was in the early 1990s, and the mechanism of gene silencing wasn't even known yet, or certainly wasn't investigated. So we really scratched our heads wondering why this one plant that did not express coprotein and expressed almost undetectable small RNA or RNA was so resistant. So we did a lot of work in that, and Michelle Rebellanandro and others really worked hard on figuring out this what the resistance mechanism was. So the next talk was by Dr. Zagrai in Romania. He's the director of the Romanian Fruit Research Station, Bistrita. Since the virus was not in the US, we could not really test for resistance in the US because in the field, because we couldn't have the virus in the field. So with the help of Dr. Rebellanandro, we were able to find researchers who were interested in doing this type of research, looking at it in the field, and we looked at it. It was looked at in Spain, Poland, and the Czech Republic, and certainly Romania. And Dr. Zagrai did a long-term study about resistance in the field. Aphid, also graft inoculation, but primarily aphid inoculation, because that's how the virus is transmitted. And he found no infection in the honey sweet trees since 1996 until today. They have never been infected, while the other control trees have been highly infected over the years. Graft inoculation, the virus could not move. It could only move a very short way from the inoculation point, never moved systemically throughout the tree. Extremely high levels of resistance. But not only did Dr. Zagrai look at that, he looked at fruit quality in terms of would this be acceptable by the consumer in terms of the fruit quality alone, and certainly it was a very good variety, and certainly in looks, in size, productivity for the grower was good. And he then did a whole series of work looking at the nutritional components, and looked at lipids, proteins, vitamins, minerals, et cetera, and found that certainly honey sweet was in the same area in terms of all these nutritional qualities as the fruit that people are enjoying in Europe at this time. So there really, it was equal to those fruit in terms of quality and nutrition. His general conclusions of all of his work to date is that that certainly there's a high level of resistance, good agronomic performance. Field trials demonstrated. Also, I failed to mention that he did look at aphid populations. He looked at bees. He looked at looking at non-target organisms. He also looked at various strains of the plumpox virus to see if there are any interactions that would change the strain composition. found it was neutral, that that honey sweet was neutral in all these effects. And his last statement was that what really is left now is the challenge now is to approval in the European by the European Union, so that growers can grow it and consumers can enjoy it. So that was his conclusion. Then we went to Chris Dardick at the USDA in Kearneysville, West Virginia. And Chris is at the same station where I retired from a few years ago. And we worked together on this project. What we did was we wanted to look more at a pan-European look at fruit quality in terms of honey sweet compared to the fruit grown all throughout Europe. And we looked at fruit from and now this is standard fruit from either from the markets or from researchers' fields. So these were standard varieties from France, Spain, Czech Republic, Bulgaria, Italy, Poland, Romania, Germany. So we, and thankfully, and we really appreciated, these researchers sent us fruit, of course, through quarantine into the US, sent us fruit so we could analyze the qualities of those fruit, the nutritional value, compare those to honey sweet. And I can just say, I can just repeat what I mentioned about Dr. Zagrai's work. Honey sweet is just as nutritious, just as high quality, has all the nutritional components at the similar levels to what we find in fruit that's commonly in the market in Europe. So that was the results of that. But we also then had fruit that was, we had Stanleys are quite a popular cultivar, especially in places where plumppox virus is endemic. And the reason is because Stanley is susceptible to plumppox virus, that this plum variety is susceptible, but it doesn't show symptoms on fruit. Therefore, growers can grow Stanley even under conditions of virus infection and market the fruit. So we wanted to see what the RNA, small RNA components were in honey sweet versus Stanley uninfected and Stanley infected. And both Stanley and honey sweet collected from the same sites where the regulated test sites in Europe. And Dr. Dardick and his colleagues did RNA-seq, small RNA-seq on a number of 36 samples of Stanley and honey sweet. I can say, you know, like 964 million reads and 200 of messenger and 200 almost 250 some odd small RNA reads. They assembled this data into, at that time, this was a number of years ago, they assembled it to the peach reference genome because there was not a plum reference genome, but since then, Dr. Dardick and his collaborators developed a genome sequence for plum, and now it's currently, the plum has a reference genome also. So what did he find out from these? And also I should mention that he did feeding studies, rodent feeding studies with sweet and Stanley. What did the messenger and small RNA studies? There were no detectable changes in transcription of flanking DNA. So at the insertion site, they looked at the flanking DNA, and there was no effect on the flanking DNA. No evidence for a new transcript that spanned the transgene plum borders. The transgene small RNAs were produced at a very low abundance. And he found that virus infection in Stanley, which is quite common, with plumppox virus. Virus infection led to more plumppox virus-related small RNA production than produced by the coprotein in honey sweet. Honey sweet produces no small RNAs that match human genes that are not already present in plumpox virus infected fruit of the Stanley, which are regularly marketed and consumed. And the rodent feeding studies showed no negative effects on a very large number of analyses, no negative effects from honey sweet feeding. Then we have Pete Vandermeer, and it turned out that there was a large number of researchers in Europe and the US that worked on honey sweet, the development, the testing. As I said, we had sites in Spain, Poland, Czech Republic, Romania. But now this group of researchers has expanded to people who are very thankfully, they're helping to look at the regulatory aspects in terms of a submission to the European Union. I should mention here that in 2011, honey sweet was deregulated by all the agencies, the APHIS, EPA, and FDA deregulated honey sweet, so it can be grown in the US, grown and fruit sold, et cetera, et cetera. Nevertheless, we don't have plumpox virus in the US. After a 10-year program, it was eradicated in a small area where it had come in. And what honey sweet is being used now in the US is not for commercial production, but it's being used as a breeding stock to develop a group of varieties, a large group of varieties that are plumpox virus resistant. The gene is transferred in breeding as a single dominant gene for resistance. It's very easy to transfer. So if plumpox virus should ever come into the United States again and start spreading, we will have a large group of germplasm varieties that will be plumpox virus, highly resistant to plumpox virus. So back to the EU, Pete Vandermeer talked about the development of an international nonprofit association called the Public Research Specialty Crops Approval Association, PRESCA. The purpose of PRESCA is to collaborate on facilitating availability of regulated crops that are developed by public research institutes for the common good. The first crop naturally that they're working with is honey sweet. But it will be open in the future to other horticultural crops. So the activities right now are preparing and submitting regulatory dossier for honey sweet. Also collaborating with research groups and other organizations, organizing workshops, conferences, collecting, analyzing, exchanging, comparing, and disseminating information, and facilitating public sector research and development of genetically modified crops. So this is PRESCA. It's getting started now, and we hope that you all will be interested in joining PRESCA and becoming part of this organization that will really, the honey sweet has no commercial, there are no commercial strings attached in terms of profit. None of the researchers are getting any profit at all, and is really being done for the public good. And we hope that it will be an example for what can be done for the public good in agriculture, to improve agriculture, especially in times of climate change and in times of the exotic organisms traveling, invasive species traveling throughout the world. Okay, so then Pete Vandersmith talked about several different ways of placing GM crops on the market in EU. He talked about several of them, but in the end, for honey sweet, the choice is going to be separate consecutive submissions. The first submission of the cultivation dossier will be followed by then a submission to the food feed dossier. So it's a dual submission. So the last speaker was Nick Vandergooy. He is also providing volunteer assistance in compiling this scientific document. And Nick has a huge task in front of him with 25 years of data. You know, after it was approved in the US in 2011, we never stopped doing research with it. So there's 25 years of data, much, I would say most of it published in referee journals. Nevertheless, to put that all together and in terms of some of the dossier will include general information, nature of the GM plant contained in the product, information related to previous releases, and information relating to the monitoring plan. So that's Nick is working with us all. There's also a large group now of people who are helping in terms of getting this through the EU system. And I really appreciate their help. So that's what I wanted to tell you all. I apologize so much for not having been able to get this on, but I know I think it will. Bruno will be able to get this onto hopefully onto something that you all can see. So I thank you all for your patience, and I thank Bruno for having this set up and giving us the opportunity.
Okay, thanks very much, Ralph. I don't see any questions to Ralph, so thanks for that. Yes, so I remind everybody that all of the presentations today will be available through the iPlanta website. And of course, all the previous webinars that have been referred to today are available on the iPlanta website. So if you want to go back and pick up more of the detail, you're very welcome to do that. So thanks very much, Ralph. And now we'll pass on to the next session. This session will be organized by Professor Hugh Jones from the University of Aberystwyth, who is also a working group leader in iPlanta. And he's going to trigger a general discussion on the future vision for RNAi in relation to the new technologies required for sustainable agriculture. So Hugh, I hope you're ready, and I hand over to you.
Okay. Thank you, Jeremy. So what we intend to do now is to kind of broaden the discussion into how we see sustainable agriculture going forward, and how all the techniques of RNAi that we've been discussing throughout this action may be used. Are you seeing my screen?
Yes, that's fine.
Full screen. Put the full screen.
Can you go to full screen? That's
Okay.
it. Yeah, okay. That's good.
So I will bring a speaker in, a good friend of mine, to kick off this session. But before I do that, what I'd like to do is just to share with you a document, a publication that just came out earlier this year, written by the US Academy of Sciences and the UK Royal Society. And it's here, it's called The Challenge of Feeding the World Sustainably. And I just want to highlight a couple of the key statements it made in its summary. So the world's population is still increasing, requiring more from our agricultural systems. Malnutrition and diet-related illnesses are present in nearly all societies. At the same time, agriculture plays a significant role in some of the biggest environmental challenges that humanity is facing, i.e. agriculture is causing these challenges. Climate change, biodiversity loss, deforestation, and the pollution of our soil, water, and air, and so on. The need to balance the growing demand for food with these environmental threats is a complex issue, and ensuring sustainable food systems will require a collaborative effort from many different communities, and of course a range of science and research. So I will just introduce the speaker, Professor Toby Bruce. Holds the chair in insect chemical ecology at Keele University in the UK. And apart from his broad research and teaching interests, he's also found time to convene the AAB's biocontrol and IPM group. He holds various visiting professorships. He's the editor-in-chief of the Journal of Physiological Entomology and the co-editor of the Annals of Applied Biology. And he's also a key member of the Keele University's Centre for Food Security. So with that, I will stop sharing my screen and ask Toby to, how do I enlarge this? Or I've shrunk the Teams window down so small that I can't see. Oh God, I can't get to my. Sorry, Toby, I'll stop sharing. Well, just see if you can share your screen over the top of mine. I'm still trying to get back to the Teams.
If Tom share his screen, he will go up on your.
Thank you, Hugh. Thanks for the kind introduction. I'll just see if I can share my screen.
If you've miniaturized your.
On the top of the screen on red, you.
Yeah, in the top right-hand corner, you should see a miniature screen, and click on the corner of that.
That is the red button.
Mm.
But okay, Bruce is coming. Perfect.
Okay, we got Bruce.
Okay.
That's it. Good.
Hello, everyone. It's really nice to have a chance to talk about some of the bigger picture issues with this. So it's been really interesting to hear your webinar about the RNAi technology and the opportunities with that. But now we're going to consider why we need innovation in crop protection in the first place. So this brief talk is just very much an overview, but the ways in which new opportunities can be created to reduce vulnerability of crops to pests. So in wild ecosystems, natural plants, they've evolved ways of withstanding attack from insects, herbivores, but in agricultural systems, it's a very different situation because it's all managed by humans, and the crops are artificially selected instead of being naturally selected, and they may have lost some of the resistance traits that exist in the wild plants. There's all kinds of ways in which the cropped ecosystems are different. So the plants are often grown outside of their area of origin. They're grown in very dense, uniform stands, and they have much more frequent disturbance. So they're very unnatural kind of ecosystems, and to have such a concentrated resource, it really creates an ideal habitat for an adapted attacking organism such as an insect pest. And that the food resource, which is useful for humans, is also something which they like to attack. So in summary, agricultural environments, they're very simplified and vulnerable to pests. These lush monocultures of artificially selected high yielding crops, they they've been often grown in a pesticide-treated background, and the pesticide has actually been part of the package during which the high-yielding varieties were developed. So we have from the Green Revolution the wonderful high-yielding varieties, which was a very remarkable achievement. But if we don't have that crop protection, then the high yield is not realised, and there's a great risk actually. I think policymakers have underestimated quite how much of a threat it is to have the crops being wiped out by an attacking pest. So just briefly, this flow chart shows some of the reasons why there's problems with pests in agricultural ecosystems, but a lot of this is driven by there being fewer effective pesticides nowadays. So this is due to the combined effect of the evolution of pesticide resistance together with much slower development of new pesticides, because the legislation is much stricter now. I mean, for good reasons, you need to have some regulation, but probably the pendulum has swung too far, and there's actually more regulation than there would be required to have the actual good balance between the benefits and the risks. So these are the challenges, and these crop systems are very vulnerable to pests, especially when there's been broad spectrum pesticides being used that may have killed the natural enemies of the pests. We've got a number of serious pests, in this shows the pests which have been reported by farmers in the UK, and the ones which are higher up in the list, these are ones where there's big problems with pesticide resistance, or where there was a previously effective pesticide which has now been banned. So things like the cabbage stem flea beetle, which became a problem after the neonicotinoid restriction. So new options are needed, and there's a real urgent need for innovation system to create new options, and this starts with research. This challenge means there's also opportunities such as the RNAi, and so we need to find new ways to make the crop environments less suitable for the pests, how to make plant resistance to pests improved, and also increase the activity of natural enemies of the pests to make the crop habitat a more favourable environment for them. We need new interventions such as the resistant crop varieties, more use of natural enemies of pests, cultural methods to reduce pest infestation and the vulnerability of crops to pests, and monitoring systems to forecast risks to crops and rationalise the use of interventions for dealing with the pests. And so this needs to be done as part of an integrated system. But the big issue that's currently being faced is that the pests are evolving resistance to the control interventions at a much faster rate than the new ones are being developed. And so there's a real urgent need for new solutions to be developed. So this is the crop protection challenge. We got this vulnerable environment, which is for the last few decades, it's been protected with cheap, readily available pesticides, but now there's a reduced availability of that. So we need research to develop new ways of controlling these pests, or otherwise there could be a situation where you get the exponential increase of these pest populations in the environment which is so suitable for them. I'd like to just go back and revisit some of the things which were said by Rachel Carson. So this is going right to the foundations of the environmentalist movement, and I think it's quite encouraging to see that the things that she actually wrote are actually much more supportive of science than some of the actions of, of some of the environmentalists, which have been more against science. But Rachel Carson, she actually saw science as being part of the solution and not part of the problem. So we're needing to have better science to develop new ways of managing the pests. So she had a vision of having entomologists, pathologists, geneticists, physiologists, biochemists and ecologists all working together to develop new solutions. And it'd be interesting to see if she was alive today, what she would think of the RNAi and whether that should be brought forward and promoted. And I think that she probably would have been very much in favour of it, actually, if it provides a better alternative to the use of broad spectrum toxic pesticides. Another sort of big picture consideration, if we really zoom out from this, is that if we have crop losses to pests, then agriculture is less efficient, and more land needs to be used to produce the same amount of crops. So that means that there's a risk of agriculture spreading sideways to meet the demand for food. So there could be more deforestation and use of wild lands, that land which is used for nature becoming used for agriculture instead, which obviously would not be good for the environment or good for biodiversity conservation. So the Royal Society in the UK came up with a landmark report in 2009 called "Reaping the Benefits," where they made the case for the need for a sustainable intensification of global agriculture. So where we can have improved yields, but without more environmental impact and without the cultivation of more land. So this is actually one of the biggest challenges that humanity is facing in the 21st century is how to do this, how to meet the rising demand while at the same time not using more resources. And there's a need for a second green revolution that relies more on knowledge than on just pouring on really high levels of inputs to get the high yield. So we need smarter solutions such as the RNAi. So this is in the context of human population growth and the need for more food, but it's not just that. There's also the consumption per person has gone up. So there's more intensive diet. So when more meat is eaten, then more food is needed. So there's demand increase. There's climate change. There's the threat to biodiversity. So we need to make sure that we're not having agriculture using more land. There's also huge issues with water in the 21st century. So agriculture, it's estimated about 70% of human extracted fresh water globally. And so we also need to think about how to use water efficiently. And if there's a lot of crop being lost to pests, then that's not going to happen. So there've been a couple of recent reviews looking into future crop protection. So there's this European one from the European Parliament, and I'd really like to highlight one of the questions which this very recent report has raised, and it says the key question arises as to whether the use of plant protection products can be reduced while maintaining or increasing yields. So that means, is there going to be a yield decline? So with the current strategy for reducing pesticides, then the likely consequence of that is that there's going to be a decline in yields, and so that's going in completely opposite direction from this sustainable intensification, which is required to meet some of these bigger picture challenges which humanity is facing. So we need to have new solutions, and there's a number of them. I think the consensus of opinion is that we need to have an integrated approach, so not relying too much on any one thing to avoid the evolution of resistance, but the kind of things that can be done, there's mechanical techniques, there's plant breeding, there's biocontrol, induced resistance. These are the things which have been mentioned in that report, but they could also include RNAi plant production products amongst this. Similarly, in the United States, there's been this Committee on Agricultural Science and Technology review paper, and they've looked into crop protection in the future and how that may contribute to agricultural productivity. They made similar points that integrated approaches are needed. That even though there's been an increase in pesticide use, crop losses have not significantly decreased in the last 40 years. So there's a real need for innovation, and they actually highlight some of the genetic techniques such as CRISPR-Cas, RNAi, marker-assisted breeding, and these various advanced techniques, and say that it could fit it well into the integrated systems. I know the current audience is very interested in RNAi, and so it's interesting to see that report has actually looked at the increase in numbers of publications and patents, and so it seems, as you will be well aware, this is a growth area, and this is one of the opportunities actually. So if we've got the challenge with the conventional pesticides, there's an opportunity to develop new things like RNAi, and there's a need for this innovation to provide new solutions, because at the moment, the farmers are actually really suffering from having a lack of tools in the toolbox for pest management. The GM crops, they got huge potential. So this is a slide from Jonathan Jones at the Sainsbury Laboratory. He developed these blight-resistant GM potatoes, which have got a gene taken from a wild potato, and they don't need all the fungicide application, but with the problems with GM crops, that's not available. But it would be nice to, just looking purely from a technical point of view, this is a really good solution, and it's a pity that for other reasons it hasn't yet been possible to use this, but there is opportunity. And so things like biopesticides, biostimulants, there's huge growth in that. So while there's challenges with current toxic pesticides, and there's fewer newer conventional pesticides being developed, there's increased development of new alternatives, which could include the RNAi. So just to sum up with now, research can increase options. It allows us to see what's feasible, to assess the risks and the benefits. So I mean, actually, a large part of the research is to weigh out those two and see whether the benefit is worth the risk. And so if the risks are considered minimal and the benefits are large, then it's something to consider, and it allows innovation and new options to be developed. And we urgently need this actually for crop protection. There's an urgent need for developing new methods. It's analogous to the situation in medicine where there's antimicrobial resistance and the need for new drugs. It's the same thing with crop protection, with all the pesticide resistance and the need to develop new ways of managing the pests. So just my final slide now. So there's all kinds of hurdles to get through to allow this research to lead to new solutions. So first of all is getting the funding for the research. Then there's also making sure it's relevant research, which is addressing the real world problems. As there's been quite a lot of discussion already today, there's issues with regulation. So, can we actually be allowed to use the outputs of the research and translate that, make it available to the farmers? Finally, is it something which the farmers can use? So it'd be good to have interaction with them early on in the process to make sure that it's something which is suitable for their needs. But if it's something which works very well, then I don't think there's going to be any problem with grower uptake, because there's a great demand for effective solutions which are going to make their lives easier for managing pests. So thanks very much for listening to the talk. It's been a great honour to be able to talk to such a special audience. So thank you very much.
So thank you, Toby. Excellent. You've clearly described the significant challenges faced by agriculture going forward, and you've outlined the urgent need for research and innovation, and the role that RNAi could play as in part of an integrated solution there. Okay, so I'll open the floor first of all to questions. So has anybody got any specific comments or questions to Toby on the things he said? And while those are coming in, I also want to involve everybody in a more open discussion about how, if at all, but I think there is obvious opportunities for how RNAi in its broadest sense can be part of the solutions to the challenges faced by agriculture. So first of all, any questions to Toby?
Well, if nobody is starting, I have well, just a comment. It was really impressive, the slide that you were showing about the amount of new biopesticide and biostimulants are now coming up and so on. In my opinion, I would say that many of that new molecular, probably, I don't know, everybody's putting a lot of effort on this, but I'm not sure that really all that molecular will be able to solve the problem that we are having. Vera was showing in her presentation what, for example, the representative of the major Italian apple association growers were saying that by taking off 3 active compounds like Captan, Mancozeb, and so on, that will affect the entire production with the risk of the loss of minimum 40% of the production. So with really a strong impact in for example, in that specific agricultural sector that in some area in Europe for apple production is really important. So that is crazy how the European Union Green Deal is forcing this without really providing solution, alternative. I don't think that among the long list of compounds that you are showing in your table, new biostimulants or biopesticide will be able to replace, to be so effective how they were effective that molecule. So what will happen in your opinion?
Yeah, that's a very good point, because you need to have some product which is going to be very effective. And at the moment, the way I see it is, there's quite complex legislation, but with the Sustainable Use Directive and everything like that, then on the one hand, they're trying to reduce the perceived risk to human health and the environment with the conventional pesticides. I think even with the conventional pesticides, if they're used responsibly, there's benefit from them. But there's a great campaign against them, and they want to reduce that. So it's a bit like a seesaw. So on the one side, that's going down, and on the other hand, in theory, there's promotion of IPM and use of alternatives, which is what it says. But I think the reality is a bit more like a broken seesaw, and it's going down at both ends. So there's the part about restricting the conventional pesticides, that's happening quite fast. But the speed at which the alternatives are becoming available is not happening fast enough. And so it's and then the farmers get caught in the middle, because they've been relying on these pesticides to protect their crops. And if they don't have it available anymore, then they get stuck in a very difficult situation, because their livelihoods depend on it, and also producing food for everyone else. And so there's some major problems. Just take a couple of examples. There's the herbicide-resistant blackgrass weed, which is making it difficult to grow wheat as a winter crop in large parts of the UK. And then the other major broadacre crop is oilseed rape. And since the neonicotinoid restriction, then it's been really hard to establish that crop, and quite a lot of farmers abandoning growing oilseed rape because these flea beetles come in and they eat the young seedlings. So we're talking about conventional farming. It's not just organic farming, and they can't actually grow a couple of the major crops, wheat and oilseed rape, due to problems with crop protection. So there is an urgent need for new solutions.
But this is an important message. We should give this message, or we should wait that apple disappear from the market. So, and then the consumer realizes there is a problem.
Yes, I think we should try and get that message across wherever it's possible. I mean, I have tried with the UK Parliament. I've been to see their Common Science and Technology Committee to try and make them aware of this, and try to give information about it, because I think the policymakers they're not aware of quite what a difficult problem this is, because I think there's a sort of widespread misconception that, you know, if we just all went back to nature, everything would be fine. But as I tried to show in my presentation, that farmed ecosystems, they're not really natural, and so if you let it go back to nature, you'd actually have all the pests and weeds and diseases taking over. And so it's how to sort of maintain this artificial situation where you've got a lot of one crop being grown for food. So yes, I think we need to try and get that message across, but to try and emphasize that there's an opportunity as well as a challenge, because we don't want to be too sort of pessimistic about it, but because this is actually something where the excellent science in the EU can make a difference, because you know, if we have the kind of new approaches that you've been talking about today, then and that you've been developing an iPlanta, then that could help to solve some of these problems. So it's actually it's good news, but we need to make sure that the innovation system is fit for purpose, and it's going to actually allow the development of these things. Because if we have such a risk-averse approach, then you never actually manage to make an improvement, and you sort of it becomes a blocked progress. So with this precautionary principle, I understand it. But if you take it to such an extreme level that you never do anything new because you're worried about what's unknown thing there could be about it, you're never actually going to be able to make any progress. And so we'll just be stuck with the same conventional pesticides, which aren't perfect themselves. They're actually very broad spectrum. They kill beneficial organisms, and so we need to have something which is more selective. And if the regulatory system doesn't allow us to develop that, then we're just going to get stuck with the status quo, and that's not a good situation either.
There are some questions here.
Yeah, okay, I'll take a look at those. I'm looking at Michelle's. I don't quite understand what Michelle is saying. Are you able to speak, Michelle, and explain what you are saying there?
I think it looks like the question's about whether it's commercially ready, RNAi.
Okay, if that's how you interpret it, Toby, answer that one. Yeah.
I think there's a need for development. It depends. It depends which pest and all the details of it. But I think this is something which is really worth pursuing. It might actually be a question that would be better answered by one of the industrial partners that you have in your consortium. You know, because as I'm mainly an academic myself, I think it would be good to get an industry view on that. But I think that you know, there's a demand for new approaches, and if it can be made effective and cost effective, then I think there would be a real demand for that.
Yeah, thank you. And of course, there are already applications of this on the market. You know, the maize corn rootworm application is out there. So you know, there are examples already in cultivation. Jonathan talks about here, Jonathan Brown, is there any research on how policymakers can create the right regulatory balance to support RNAi innovation with feedback from farmers and environmentalists?
Could I make a comment on that, Hugh?
Yes, please, John.
Yeah, there have been moves to try and broaden regulatory systems, both in the UK and in Europe, to look at, first of all, to look at benefits, but also to look at how new products can fit into sustainable agriculture and into IPM systems. So there is a move not just to, you know, look directly at the product and to see whether it's toxic or not and acceptable from that perspective, but to say, okay, we've got this product which looks as though it's fairly safe, but what will happen when it actually gets used in agriculture? What will it do to biodiversity, agricultural systems and so on? And can it make a useful, sustainable contribution? And also, I think now with carbon being more as a feature, you know, what does it do to the carbon balance? Does it increase or decrease release of atmospheric CO2 and so on? So there are moves to try and broaden the regulations to look at these things, but it's going to be, I think, a while before this happens, and it's going to be quite a difficult thing to do because you know, how long is a piece of string? How far do you go in looking at benefits, disbenefits, impact on sustainability in relation to socioeconomic benefits and who benefits and so on? So, I mean, these discussions are ongoing, but I'm not quite sure, you know, what will happen and how they will materialize.
I think it's an important point because the consideration of benefits seems to have almost been forgotten about, and there's just so much of a consideration of risks. But with other things in life, even with simple things like deciding whether to cross a road or not, you've got to weigh out the costs and the benefits. It's a bit of an unusual situation to say, well, we can't do anything because we're so worried about the risks and almost not consider the benefits at all. So I think it's an important point that's been raised there.
I think this comes back to the comment I think that Hugh made earlier about the sustainable intensification that was discussed by the Royal Society and US Academy. That obviously the concept of sustainable intensification is that we increase production, but we don't do it in a way that puts more load on the environment.
Yes.
And obviously, you know, how we do that and how we assess that is quite important. So I think it all fits within that, but obviously to get that into regulatory systems is often very difficult, particularly when it's not black and white. You know, regulators like to have yes, no answers, and the answer will be maybe or yes, it could, if managed the right way, etc., etc. And that's going to, I think, make life difficult for regulators to accept.
It's true, but we need to try and find some way to expedite the process, because as you mentioned, these challenges, they need new approaches to improve the situation, and so we need to have access to technology to do it, and if they're not going to accept technology with any amount of risk, then it's going to be very difficult to make any progress. What I find very surprising actually is the way there's with the conventional breeding, we have the mutagenesis screening, where you subject the seeds to high doses of radiation and mutagenic chemicals to create random mutations, and this is part of conventional breeding, and no one's really too bothered about that. And then you have other things like site-directed mutagenesis, and that's not allowed. And the boundaries seem to have got blurred with some of the new things that can be done now, and it just doesn't seem at all logical, some of the things which are being done. So there's for things, I know it's not RNAi, but it's a similar sort of genetic technology where you've got the site-directed mutagenesis, and that could be used for crop genetic improvement. But at the moment, it's being considered like a GM crop, this gene editing, but then at the same time, you know, you could do random mutagenesis, and it has been going on for I think about 100 years now, and those crops have, you can grow them even on organic farms. So it just doesn't seem to be at all logical.
Okay, so there's a question from Hildegun about how do we communicate the challenges that you've described when we don't see changes in the prices of, let's say, oil seeds or wheat in the stores?
Yes, that's would be the bottom line, actually. So if the prices went up, it would change attitudes, and I think actually that it could be a situation where the modern consumer has actually been a bit spoilt, because we've never had such low prices for food in relation to people's salaries in other periods in history. It's historically low, and that's partly as a result of the developments with science in agriculture. But the trouble is, though, that with a particular area, or even a geographic area as big as the EU, we're in a global market, and the supermarkets that are selling the food, they can import globally. And so even if a few small farmers go out of business somewhere in the UK or the EU, it's not going to make a difference to them, because the food prices are still going to stay the same, because they'll be importing food from some other part of the world where they don't have quite the same restrictions, and where they have more options available. And so it's and I think that's why the prices are not going up because of this, because we just, you know, if you can't grow wheat in the certain parts of the UK, for example, then you'd just be importing it from somewhere else, and so that's why the prices aren't changing, and so that actually makes the communication of these issues a lot harder.
Yeah, that's true. Okay, so Ralph reminds me rightly that, of course, honey sweet plum is almost it's available commercially. I think there is still just some small hurdles to go, and it's had a lot of research to support it. Livio Trenotti asks how you comment on the increase in the use of microbes as biopesticides versus the downward trend of single molecule chemicals.
Yes, that could be something where you have less of a risk of resistance. So if you've got single molecule, there's intense selection pressure for resistance. But as with all of these things, it's quite incredible how these pests manage to evolve resistance and find some ways around whatever you throw at it. So I think that, you know, to avoid resistance problems, it's good to have a number of options, and that's actually why there's a problem at the moment, because there are not so many options available. But I think also this increase in having more biologicals and fewer of the traditional toxic chemical pesticides is driven just by the difficulty that there is in registering a new chemical pesticide, because the cost of registering it costs 100 million euros or more, and then it's just a matter of time before resistance evolves, and so it's not such an attractive
Okay, thank you, Toby, for those insights. There appears to be no further comment, so can I hand the session back to Bruno?
Sure. Yeah. Thanks for the discussion. Thanks, everyone.
Thanks, Toby.
Thanks to you for sharing this discussion, and you for your interesting points and response also. Well, I think that remain an open issue, and I'm glad that thanks to this cross-section we were able to raise many of these points, probably not giving any solution, but in some way make people discussing, and maybe making people more aware of the fact that we have, as you defined, a more smart solution that is the RNA technology. So we need to keep training people in understanding what is this technology, and we get many messages, many information, and I think that for us it will be very helpful. And let's hope that we can continue with this discussion. Unfortunately, iPlanta officially now is practically finished, so we will not have any other possibility to continue it directly. Maybe we made an attempt for having additional here, but we are not sure that it will be approved. In case it will be approved, we will continue. But I think that what done in the last 4 years and a half were really interesting. It was really interesting, and helped a lot to create a really large community, a really large network. So I really hope that in some way we will be able to maintain this network alive. Maybe iPlanta alive. I don't know yet how, but in some way we really hope that all this collaboration, will bring together more time with people for more time and for new activities. And let's hope that we, in any case, we were able to give a contribution for the development of these new technologies that we believe, we are convinced that will have an impact for the future of the agriculture in Europe. I think that Europe really needs this new solution. And so I hope that people will take in consideration all this. Well, I don't know. We need to finish. I want to thank all the working group leaders that were really, let's say, the strong support, because I'm still stressing them in sending documents and information. We need to complete the project with the final reporting, and tomorrow with the final management committee. So please, who is connected today, representing the different country, signatory country of the memorandum of understanding of iPlanta cross-section, it's important to have at least one from each country connected tomorrow, so at least we can have a general final management committee meeting and approval of all the activities that were done. And beside all the other people, I also thank all the participants. We had really many participants. Also today we reached more than 100 participants. So I think that even all the bad restrictions that we had for the COVID pandemic, I think that finally the solution we found with the webinar was really positive to keep and even enlarge the network. Unfortunately, yes, we missed to get together. The final, I want to remember and thank also the Spanish people, because the final conference was planned in Valencia, and they were ready to host the meeting in Valencia, and we were all happy to go to Valencia, but it was not possible. And so let's hope that in the next future there will be really new opportunity to have other meeting in presence, and maybe start again with some new collaboration, starting with the meeting directly in Valencia, in Spain. That is always a nice place to go. So that's all from my side. I don't know if you, Jeremy, or also the working group leaders want to say a few words to everybody. And also, once we stop the webinar, we thank everybody.
Could I just make a quick comment, Bruno? Two things. First of all, we have been talking about research, and I remind people that the new Horizon program is coming up fairly soon. I think the calls open in April, and there will be opportunities there for applications in relation to sustainable agriculture, the Green Deal, and the farm to fork strategy, and so on. So maybe people like to start thinking about that, and maybe we could put together a research proposal which relates to RNAi. So that's something to think about. The other thing is to basically, for the people who are not able to come to the MC meeting tomorrow, I think you would all like me to thank Bruno for organizing this iPlanta cost action. It's been a very successful action, I think largely because of the time and the effort that Bruno has put into this and his organizational ability. When I was working at EFSA together with Salvatore and some other colleagues, we used to joke about the national characteristics. For example, how the English were experts at culinary productions, and how we always used to say that Joe should prepare the food or the menu for our next meeting. And we used to joke about this. And of course, Italians tend to get labeled as not being good organizers. My experience has been that Bruno is an example that goes completely against that. He is a fantastic organizer, and it just shows that there are, we shouldn't be painting people with brushes and labeling them according to their nationality. So thank you very much, Bruno, and for organizing all of this and for all the hard work that you've done. No doubt we will be keeping in touch with you over RNA and other issues in the future. But I do remind people that they must come to the MC meeting tomorrow because we need a quorum, and maybe there'll be a bit of time to discuss possibilities of further research collaboration and other initiatives that are going on. So thank you.
Thank you, Jeremy. So I think that this sources of it was the result of this kind of interaction. And sorry again if I've been stressing you a lot, and I've been stressing all the other people. But I mean, I don't know. I don't want to consider with a German or UK attitude. Sorry, but more specialized on this. I don't know if there are any other comments, or otherwise please turn on your camera. So please remember that we always take a final photo. And so
Jonathan Brown.
Yeah, hello. Maybe also if there are some feedback also from some of the people attending the meeting. Because many people were following us in silence, and so there is time for some feedback from somebody. Meantime, we are waiting for people, Lorenzo, Olivier, and Toby. I don't know if they are there. We need to get the Lorenzo. I think that let's wait a minute because there are some. Let me take more picture. This is one. This is another. And this is another. Okay. Okay, thank you. If no other feedback, comments, thank you very much. And have a nice.
Can I?
Thank you. Goodbye.
Thank you. Ciao.
Bye to everybody. Maybe.
Thank you. Bye-bye.
Bye to everybody.
Bye-bye. Bye. Take care. Bye-bye.
Thank you. Bye.
Bruno,
Yes,
can I ask you a question or a suggestion?
yes.
So I'm in Ontario, Canada,
Oh, okay.
and we're looking at sustainability through the lens of the Sustainable Development Goals, the UN. And we're trying to get students involved. And I know that with Europe and the new Green Deal, there's a fantastic opportunity to connect up collaborative research projects with post-secondary students in K to 12 through hackathons or what have you. But have you looked at that opportunity in Europe in terms of the Erasmus program or some of the other programs that connect up students with the research and also with the